So now we know how to quantify a fluorescent signal, and in principle also know how to do this in a microscope. You've seen a setup for a fluorescence microscope in one of the early lectures. We still don't know how to label molecules specifically with extrinsic labels. In biology, we nearly always need to do that to get specificity. And the technology to do this has matured enormously over the years. And in fact, the making of dyes and the creation of technologies that allow us to look at molecules with good sensitivity and specificity in living systems has advanced biology enormously and biotechnology. And so in fact, the invention of one of these techniques that we'll briefly touch on today, the one of fluorescent proteins or fusion protein technologies, has won 3 scientists a Nobel Prize in 2008 in chemistry, in fact. But actually the science behind this is a classic biotechnology and molecular biotechnology example. And so in this lecture, I will just give you an introduction to this vast topic. And if you want more details, then there's excellent literature out there. And also, if you want to consult the web, just Google School of Fluorescence in the search box, and that will lead you to the Molecular Probes website. And with that link, you will find really good explanations on how some of these technologies work and how they work in more detail than I'm going to talk about it here. So if you do research, please consult those pages. In fact, some of the things that I'm talking about here make reference to the site there. So the key concepts we talk about here are that we use organic fluorophores that can be functionalized in such a way so that they bind to specific targets. So that's what we do in this lecture today, lecture 12. And we want organic fluorophores because of the multiple conjugated bonds that we encounter there, and that makes them good dyes and strong dyes with high brightness. We will talk about the concept of immunofluorescence, using antibodies to target proteins inside cells. And we will talk about 2 different types. One is called direct and the other indirect immunofluorescence. And we will see what that means and what the advantages are. And then finally, I just alluded to this, the fluorescent protein technology, which makes use of so-called recombinant DNA to create a fusion construct between the protein that you're interested in and another protein that actually is the fluorescent reporter. And this is a great advance because it now allows us to look at living processes with high specificity. So I referred to this website already down here, Molecular Probe School of Fluorescence, if you want further details. Okay, so a few key properties of organic dyes first. So one, or fluorophores in general, important is the brightness. The brightness is a combination of 2 things, as you can imagine from the last lecture on rate equations. You require both a good absorption coefficient. So the absorbance, the fractional absorbance is determined by the molar extinction coefficient, as we've seen from Baeyer-Lambert's law. So let me just point to this with the laser pointer, and yes. So the one part of the brightness is how well photons are absorbed, and so good dyes have extinction coefficients epsilon of between 10,000 and a few 100,000 per mole cubic decimeter per centimeter. And on the emission side, you need a high quantum yield. So you want that the energy you're putting in comes out as signal. And so for that, you want quantum yields that are at least 10%, but can reach for some systems as much as 90%. So a quantum yield of 0.9 means that 90% of all the excited molecules will actually give you a signal photon. And that's achievable with good dye design. The color is, of course, important. You want dyes in the microscope to have clearly separated excitation spectra and emission spectra. Their colors need to be well separated so you can get good signal to noise ratio. And if you want to label multiple species at once, then you might be able to distinguish them by using different color extrinsic labels. And so making sure that they don't overlap is super important. Just to give you an example here on the on this figure 1a, you see the spectra of a number of dyes that can be used in biological imaging, and you see the emission spectra here. You see spectra ranging from the blue in the 400 nanometer range right down to the red in the 750. So this is getting towards the near infrared region. So across the whole visible spectrum, you can get a whole bunch of dyes that if you use filters critically and in an optimal fashion, allows you to distinguish several dyes at once. But you can see one of the problems that you always face when having multiple dye labels is spectral overlap. So you can see that each of these spectra is bleeding into some of the adjacent spectra. And so your ability to discriminate it rests on your ability to really distinguish clearly between two different colors. So the Stokes shift is super important. You want large separation of excitation and emission so you can differentiate. But then there are very different types of chemical and biological properties that the dyes must fulfill. I mean, it must not be toxic to your system if you look at living systems, but they must also be small often, because if you have a target molecule and the dye molecule is larger than your target molecule, that can cause problems in the functionality of the labeled molecule. So you don't, if you want living systems, you do not want to change the properties of the labeled molecule itself. In some cases, it doesn't matter if you have to kill the system beforehand before you image it. Then, of course, this is not so much of a criterion. But others are water solubility, so the pH sensitivity of the fluorescence properties. So if you have a system where you have a large variation of pHs, then the intensity itself will not only tell you how many molecules you have there, but it will be dependent on the pH. You may or may not want that. It depends on the question that you have. So if you want to report on an environmental parameter, that's one thing. But if you want to report on the concentration, you need to take care of these things. The photostability is super important. If we pump lots of photons in and out of the system, as we've seen in the last lecture, then perhaps after a couple of cycles the molecule disintegrates, and so that would be that that loss term that we didn't include in the rate equations in the last lecture, but that I mentioned might be there. And then finally, very important is the functionalization of these dyes. How easily can you take these dyes, these dye molecules, and can you derivatize them in such a way that they become they become attachable to the target that you seek to label. So just to come back to this Stokes shift, so what you see down here is a a dye molecule that has this this excitation spectrum, so it will absorb over this range here, so roughly between 350 and 400 nanometers, and then it will emit somewhere around here from 400 to 500 nanometers, whatever that is. And what you want is a clear separation between emission and excitation, so that you can use filters that will pass what comes through here and you know, will clearly distinguish between the excitation and the emission. And so you've already seen how you do that. So you may have your light source, then you have this excitation filter. We've seen this in the past. The excitation filter will pick out the color band that you want to excite the particular fluorophore you're interested in, pass that light down onto the sample. The fluorescence, the Stokes shifted light, passes through this dichroic beam splitter, so it's what's called a long pass filter. So you can see it has this sort of spectral characteristic. You choose it so that it rejects the excitation light, but it transmits the emission light. And so you then pass it through a filter that's shown sort of in this shaded blue region here. So part of the light will be transmitted as signal. And so choosing these filter sets carefully is super important in any practical experiment, so you can be sure that the signal stems from the molecule that you're interested in and not something else. Okay, so let's look at how we make dyes functional. And one of the gurus that you've already met is this guy, Gregorio Weber, who really invented this field of immunofluorescence and dye staining of biological molecules. One example of a dye that is functional just by virtue of its geometry is a DNA binding dye, which is called DAPI. And so what you see, it contains these doubly conjugated, multiply conjugated rings, aromatic rings here, and they're arranged in this plane. And there's a single axis that connects this ring structure with this ring here, and this dye molecule has a shape that seems to fit really neatly into the minor groove of double-stranded DNA. So you can see this here. So here's a DNA strand, and you see the dye intercalating into this strand. And the nice thing is when it fits into this groove, the DNA stabilizes the dye. And so essentially this dye now doesn't rotate and wobble around this axis here. And because it's now held stable in this plane configuration, these electrons can now delocalize really easily across this entire molecule, and it becomes brightly fluorescent. So this intercalation, this shape, the shape of this dye, it makes it highly affine to bind to the DNA, and the DNA stabilizes it, and once it's bound, it becomes brightly fluorescent. So in that sense, it's a functional dye that gives you strong signal when DNA is present. And so this dye, DAPI, has an excitation maximum of 345 nanometer and emission maximum of 455 nanometer, and it's used a lot to denote the nuclear region of a cell where there is, of course, a lot of DNA, and so you would get the strongest signal from that region. There are other dyes. You can functionalize dyes that have this ring structure here. For example, so here are two similar dyes. One is called NHS fluorescein. So this is fluorescein, which is functionalized with an HS group. So this functional group binds to primary amines in proteins and peptides. Or if they're present in other biomolecules, then there too. So this is really a nice thing, because then you just get a signal that's specific to the protein load, right? In a particular case. So these are such dyes. So one is NHS fluorescein, the other one is FITS, which has this this isothiocyanate group on it that does the same thing. You can also get dyes that are reactive to sulfhydryl groups in proteins. This will be in molecules, for example, that contain cysteine groups and so on. And one such dye, so this is again the actual fluorophore properties are given by the same backbone structure as you had in the previous cases here. It's just a different derivative that now allows it to bind to make thiol bonds. And so this is a way you can actually functionalize different dye molecules and different colors to make this happen. But the problem is, of course, you do this, but this may not be still very specific to individual proteins, right? This tells you there are lots of proteins present, but it doesn't actually tell you which ones, right? So for most assays, this will not be so interesting, although it can give you contrast to give you overall topology of the cell and so on, you know, because the protein content or the content where these molecules are reactive may be localized to certain particular parts of the cell. So how do we gain specificity? How can we target a particular protein? Let's say I suspect a certain protein let's call it protein A is involved in cancer, and I would like to test its presence inside a cell. How do I make the color appear, and how can I be sure that color from the reported dye really only refers to the presence of protein A and not the many other proteins that are present in the cell? So this is the field of immunofluorescence. So this is the field of targeting proteins with great specificity. Again, Gregorio Weber was super important in this field, starting this off. And so what you do is you use so-called antibodies that are produced during an immunoresponse inside the body, essentially to destroy foreign molecules or things that are in your body that it doesn't want. And so your body produces these highly specific antibodies that are shaped in such a way that they can bind the so-called antigen. That's the thing that you that they are made to target inside the body. So you're using essentially the natural cell machinery of living organisms to make these specific antibodies towards the antigen that you're interested in. So the antigen in immunofluorescence will be the protein. So going back to the previous example, so you want the antigen to be protein A, and you want to design the antigen, and you want to design an antibody that only binds to protein A, but not the other proteins that are inside the body. So the antibody binds specifically, but not covalently to another molecule, and this other molecule is called the antigen. And the site on the antigen, so the site that actually is exposed to the antibody and where the antibody actually provides the tight fit for it to bind, for the antigen to bind, is called the epitope. So that is the actual thing that interacts with the antibody. And so I will tell you on the next slide just how one raises such antibodies. If you have a certain protein, how do I make the antibody for it? I'll show you that in the next picture. But let's suppose we have them. Then there are two types of labeling the protein. So let's stick with protein A. Let's suppose protein A is that yellow blob on a cell, and I want to label it. So what I do is I take a primary an antibody, and So the simplest way of doing it is called direct labeling with a so-called primary antibody. So it's a primary antibody that has been produced to really just recognize that protein, so the yellow blob here. And you then use techniques like I've shown in the previous slide. So if you use a standard organic dye that's been derivatized to recognize primary amines, so the amines inside the antibody can now be labeled with these organic fluorophores so that the antibody itself becomes highly fluorescent. So first step is make the antibody, then label it with strong organic dyes, and then you incubate the cells with the antibody, and they will only bind to the targets that they're designed for. The rest you wash away, and now when you look at fluorescence, you know that the signal comes from the protein itself, because only that will be labeled. The other way of labeling is to raise a primary antibody, but then use what's called a secondary antibody. Now, the secondary antibody is one that recognizes the primary antibody as its antigen. So essentially, there's an epitope on the primary antibody that is recognized specifically by the secondary, and the secondary is then labeled with organic dyes, just as we had in the previous case. And so you get a rather more complex step. So it's a two-step process. First, you wash with the primary, and you bind, and then you have the secondary put on, and then you do the fluorescence experiments. You wonder why go through all that complication. Why not just do the primaries? And there is a good reason for that, and I will come back to that, because in fact this second method is much more versatile, because you do not actually have to make fluorescent primaries, which can be very expensive. So this process of labeling can be very expensive, and you'd have to do it for every single primary. In this case, you only need to label the secondary. And the secondary can be just a generic antibody that recognizes a type of primaries, and the primaries only have the binding sites modified, but you don't have to modify the secondary at all. It will always be the same one. So I will talk about that more. So for example, you could have a primary antibody that is made by a mouse against a certain protein, and then you use a secondary antibody which is made by a goat, okay, in a goat, really truly in the animal. So and what I mean by that, so this is how you might make an antibody in a mouse. Let's suppose I have a protein that has this shape here. It's this blue molecule. I inject it into the mouse, and the mouse then recognizes something in its body that it doesn't want. So there's an immunoresponse to it, and the antibodies, highly specific antibodies shown here in red, that really only fit this molecule very specifically are produced naturally in the body. You isolate this from the blood plasma of the mouse. You take that out. You isolate it from the serum of the mouse, and then you have the antibodies. And then now what you do is you attach the fluorescent labels using the techniques I've already described. And the next step is then to use these labeled antibodies and put them on the cells where you suspect this protein is present, and then you know where you get the signal. You get a signal from a region where there's lots of this protein. So this is a mouse antibody, right? And this would be an example of a primary antibody that's labeled with a dye. So you could use this in a direct labeling approach. So here you see a protocol for this. What do you do in practice now once you have these antibodies? So what you do is you take a cell, and you then fix the cell. And the reason to fix the cell is essentially you put a bit of formaldehyde onto the cell, and what that does, it cross-links all the protein structures inside the cell so that they are stable. And And because the next part is you need to get the antibody into the cell. The antibody is quite a large molecule, and the cell membrane wouldn't be permeable to it. So what you have to do is you have to actually permeabilize the cell membrane in the second step. Whether you use a weak detergent to do that, so you punch holes into the membrane or even remove it. And if you do that, of course, you can imagine that if you don't fix the cell first, the cell would just collapse, and you just have this this mess, because the stability of it is completely gone. So you have to fix the cell first, to cross-link the cell structure, microtubule, cytoskeleton, and so on, and then the permeabilization step keeps this all intact in shape. You then add what's called a blocker, because what that means is, of course, there are thousands and thousands of proteins and only a few of protein A which you want to label. I'll stick with that example. So if your antibody that you've made has a certain sensitivity and specificity, then of course it will label the molecules that you're interested in. But if there are millions more of other molecules, not protein A, then of course a certain amount of specificity, a certain loss of specificity means that you may still get a lot of stray signal that you don't want. And to prevent that, what you do is you just put a blocker on. You put an unspecific antibody on that is not for protein A. It's just a generic antibody from a mouse, so another antibody from mouse, as you've seen before. And then what you do is you just block everything, so that most sites are actually occupied. So that the likelihood that your primary that you've raised is unspecific is reduced by that process. And then you add your labeled antibody. Then you do this by direct labeling, like this. So you have the directly labeled antibody that we've talked about. It binds to your protein, and then you wash away. So where there is no protein present, there's no signal, and where the protein is, you get the signal. This is what is called direct immunofluorescence. Okay. Just raise the antibody, label it, fix the cell, permeabilize it, block it to get unspecific signal away, put your antibody on, then do the imaging experiment, and you then know that where you get the signal, you have the protein. So that's direct immunofluorescence. The other method is indirect. So here you have an unlabeled primary. You raise it in the same way as you've seen before. Let's suppose you raise it in mouse first. And then you follow the same steps as before. You fix, permeabilize, block, put the antibody on. Then you put the secondary on. The secondary could, for example, be a generic antibody that recognizes mouse antibodies. So you take, for example, a generic library of mouse antibodies, you inject it into a goat. The goat now in its blood will produce the antibodies against the mouse epitopes, and so you take those out, and then you label the secondaries directly. And so then the next step is to put the secondary on. They will bind to the primaries, and now you get the signal from the secondary. And you say, I said again, why that complication? Well, the advantages of these two methods are, so this is this method here is called indirect immunofluorescence when you use secondaries. So what's the advantage of direct, and what's the advantage of indirect, and what are the respective disadvantages? Well, the direct labeling is quicker to, the protocol of staining is quicker because you have only one step rather than two, and you know, so that's one good thing. But the biggest disadvantage of that method is that for every protein that you would like an antibody for, let's suppose you have 5 proteins you want to label, A, B, C, and D, with different organic dyes, you need to prepare the primaries and all label them with new dyes. So that is enormously costly and time consuming. Now instead, if you raise 5 primaries in mouse against the 5 targets, that you can do that. You isolate the primaries, and then you just use a generic so let's do that in mouse. Then you raise a generic anti-mouse secondary antibody in goat. Then you only need to label that secondary. And it doesn't now matter. You're very flexible at generating new antibodies in mouse. You just use a secondary that recognizes mouse, and you get your fluorescent signal without having to functionalize the antibodies with dyes. And so, of course, there are disadvantages here too. The secondaries mean that the actual preparation of the samples takes more time because you need to do two rounds of labeling. But there is an advantage often, because often you find you can make these systems such that there are several epitopes that the secondary can recognize on the primary. So several antibodies, several secondary can bind simultaneously on a single primary. And that means the signal is massively boosted. Okay, and you can read more details yourself about the advantages and disadvantages of these two methods. So on this slide here, you see a cell from African water mongoose, which has been fixed and permeabilized. And so, but before fixation, there was a dye was added called mitotracker, which labels mitochondria inside the cell. And this is shown here in red. So the energy units inside the cell, the mitochondria, appear as these red dots. You also see in green the cytoskeleton of the cell. So this labels actin inside the cytoskeleton, appearing as these long filaments here. And that's stained with a dye called phalloidin Alexa fluorophore, so phalloidin AF488, so appearing here in green. And the nucleus is stained with a dye called Höchst, and it works exactly like this intercalating dye DAPI that I've shown you earlier. So you can get really nice detail from these small cells, and so maybe this is a few tens of microns across here in size. So you can see what fluorescence microscopy allows you to do. Here you see a carcinoma cell, so sort of a cancer-like cell, and you can see tubulin here. So these are the microtubuli. These are, again, long filaments inside the cell, very important for transport and so on in the cell, appearing in red. And they are labeled with indirect immunofluorescence. So this means they're labeled with I think it was a mouse primary antibody and a goat anti-mouse secondary antibody. And then the actin is again labeled and is appearing here in green. So the actin extends out towards the membrane, so you get this sort of outline effect showing the extremities of the cell in green. So you really get nice detail, as you can see, with these immunofluorescence methods. But all of them have a problem. I mean, so all these methods require fixation, permeabilization, so these cells are certainly dead. Nothing's moving in them. There's no proper membrane anymore in there, so there can't be anything inside that's still alive. So how do we actually gain information like this from living systems? And that's where the great discovery of the fluorescent protein technology comes into play, which won these three guys, Roger Chen, Osamu Shimomura, and Martin Chalfie, the Nobel Prize in Chemistry in 2008. And the idea is brilliant. I mean, so if you have a protein that looks like this, so here's the structure of a protein shown here in this cartoon-like fashion, and what you do is if you have the gene for that protein and you have the gene for another protein, but that protein fluoresces, acts like a dye, and you get the cell to somehow fuse those together, and make a new protein, a so-called fusion protein, that contains both the target and the reporter, then you are able to look at this protein as it is produced inside the living cell, only that it is now colored. It's the original protein plus a fluorophore on top of it. The actual fluorescent protein comes from a jellyfish called Equoria victoria that lives in the North Atlantic, I think. And so the color bit comes from a protein structure that looks like this. It's really rather like a Coke barrel. So the protein structure cartoon is shown in here. So there's a protective cage of beta sheets, as they're called, so beta sheet arrangements of proteins sitting around a chromophore, so that's the fluorescent part that's inside. So you see these aromatic rings that sit protected inside this protein, and if you shine light on it, it emits a really bright color and emits it with good quantum efficiency. And so the amazing thing about this protein, this green fluorescent protein, as the original variant is called, is that it contains of a series of residues, just 3 residues that fold spontaneously in almost any environment, whether it's a mammalian cell or whether it's a jellyfish or whether it's a bacterial cell. It's remarkable. It doesn't require any machinery, enzymes and so on, to fold into the shape where it makes this fluorophore, and that's why this technology is so immensely powerful. So it spontaneously forms into this barrel structure and becomes highly fluorescent. And you can then actually make mutations of this fluorescent proteins, and that's work that these 3 Nobel Prize winners I mean Roger Chen particularly, have been amazing at. They have mutated them in painstaking experiments to actually to actually generate a whole range of colors that allow you to target many species simultaneously inside living cells. And so how do you actually apply this technique? You use recombinant DNA technology. So it's a classic molecular biology, biotechnology example. So here you see the structure of GFP. You see the chromophore part of it. The size is about 27 kilodaltons, so it's like a small protein. So it's actually relatively large, right? It's remarkable. You can attach that to a protein and the living cells just continue to work in the normal way. So what you then do is you engineer a little plasmid, as it's called. So this is a bacterial DNA vector that you can you can cleave to open, and then you insert the DNA fragment of the target that you want to clone, and so you now get the original DNA with this extra bit in there, and then it's actually you then the plasmids you can multiply in a bacterial culture, so you get lots of them. So bacteria will just do that for you. So you isolate these plasmids, so you get lots of them, and then you somehow deliver them to the cell that you're interested in. You can do that by a technique called lipofection, so little fat droplets that fuse through the cell membrane, go into the gets into the cell, and then actually the DNA bit is just read off as if it was native, and the proteins are made, and the proteins that you originally were interested in are the same and behave the same way as they did before, despite the fact that they now have this label attached to it, this fusion protein attached to it, this GFP, or in this case YFP attached to it. So here you see 2 pigs. They are pigs in all the in every sense, except this one on the left has collagen proteins labeled with YFP, so its trotter and its feet appear in yellow rather than but otherwise it's a healthy pig in all other senses. So whilst this doesn't look particularly useful, I mean, this crazily this was used for making pets in certain countries. you can use this, of course, for really important research questions in living cells. And I'll show you a few examples like that in later lectures. Also here, actually, just following this. So there are great advantages. You can use it in live cells. Usually, you can use it in a wide range of systems. The biology doesn't often seem to be affected by it, although you do need to check this. There are some disadvantages. I mean, of course, these are good fluorophores, but they're nowhere near as bright as, for example, organic fluorophores. And so they bleach much more quickly, and you need to be very careful when you deal with them. And so the great thing is that you can look at it without killing the system. So you see one example here where you look at red blood cells and in the heart of a small zebrafish, and this has been actually all labeled with fusion protein technology like that. So you see the tissue samples. Let me see. Let me just go back to I just want to turn that off. So yeah, so you see, you can do this whilst you see the heart pumping. Look at that small scale bar. This is only 25 microns, so about half the width of a hair. And you can see the individual cells being pumped through the heart. And you see what the muscle tissue inside the heart is doing. So clearly this heart and this fish is alive whilst you being able to image them with this technology. And so even more spectacular recent advances. So this is from another Nobel Prize winner, from Eric Betzig. He didn't win this for fusion protein technology, but he regularly uses it in his research of making high-resolution microscopes. So what you can see is here you can see at very fast imaging, you can see filopodia. So these are essentially the feelers on the cell where the cytoskeleton is probing. It's remodeled all the time, so causing cell protrusions to sense the environment around. So that's what's used to make cells crawl or make cells feel what sort of swims by. So it's remarkable that we are now able to see these things. You should just keep in mind that maybe 10, 15 years ago, it wasn't possible to see these kind of things, which are now becoming routine. So it really is a revolution for biology. And with that, I stop this lecture, and in the coming lectures, we will talk about functional imaging techniques and applications.